Have you ever wondered how our cells selectively absorb oxygen, glucose, cholesterol, and even ions like sodium and potassium while keeping harmful substances out? The answer lies in one of biology's most fascinating topics, transport across the plasma membrane. In this video, we will explore how molecules move in and out of cells through passive transport, active transport, and receptor-mediated endocytosis, all explained in a simple and engaging way. Introduction. Basically, the plasma membrane is the cell's protective barrier. It has two major roles. It retains the cell's contents, cell's inner contents, and it regulates exchange with the environment. The lipid bilayer prevents loss of polar solutes, but nutrients, ions, and waste must still cross it. Transport can be either active or passive transport. Active transport needs energy and moves against gradient, but passive transport needs no energy and moves down gradient. The net flux is the overall movement. Influx means into the cell and efflux means out of the cell. The image showing the membrane transport, both active transport and passive transport. Passive transport can be of two types, diffusion and facilitated diffusion. Now we'll study about the LDL receptor mediated endocytosis. LDL means low density lipoprotein. So this is a special type of receptor mediated endocytosis in which cells take in low density lipoprotein or LDL particles. LDL or low density lipoprotein carries cholesterol in the blood, and this process shows how cells can selectively absorb specific molecules from their surroundings. Steps in the process. First of all, first one is the binding. LDL particles, this is the LDL particle attached to LDL receptors on the cell surface. Clathrin coated pits, this is clathrin coated pits. These receptor LDL complexes gather in the small depression of the membrane called clathrin coated pits. This is the clathrin coated pits, and this is clathrin coated vesicle, and it has that pits fold inward and pinch off, forming a clathrin coated vesicle. Now then, endosome formation. The vesicles lost their clathrin coat and fuse with early endosome. This is the formation of endosome, fusion with endosome. And then receptor recycling. LDL receptors are then separated and sent back to the membrane, plasma membrane, to be used again. Lysosomal degradation. The LDL particles are delivered to lysosome where they are broken down to release cholesterol. Functions. This process provides cholesterol for cell membrane synthesis and steroid hormone production. It helps maintain cholesterol balance in the body. Clinical importance. If there are mutations in LDL receptors, cells cannot take up LDL efficiently. These lead to familial hypercholesterolemia, a condition with very high cholesterol levels in the blood and an increased risk of heart disease. Active transport. Active transport is the process by which cells move substances against their concentration gradient from low to high concentration. Unlike simple or facilitated diffusion, this process requires energy. Specialized proteins in the plasma membrane, often called pumps, carry out active transport by changing their shape to move molecules. The energy for this process comes from ATP hydrolysis, light absorption, and electron transport or the movement of or other substances down their gradients. This image shows the main types of transport. First one is passive transport and active transport. Passive transport can be divided into simple diffusion and facilitated diffusion, and active transport is divided into primary active transport and secondary active transport, which is further divided into co-transport and counter-transport or anti-transport. Primary active transport, this is example for this is Na plus K plus pump. In this process or in this type, energy from ATP is directly used to move ions across the membrane. Mechanism. The Na plus K plus ATPase pump moves 3 sodium ions or Na plus out of the cell and 2 potassium ions or K plus in for every ATP molecule broken down. This creates a high sodium, high Na plus concentration outside the cell and a high K plus concentration inside. As a result, an electrochemical gradient is formed. Significance. It helps maintain osmotic balance. It maintains the resting membrane potential in cells. It provides the Na plus gradient required for secondary active transport. Secondary active transport, that is Na plus glucose co-transport. Here energy is not used directly. Instead, the process relies on the Na plus gradient created by the Na plus K plus pump mechanism. The Na plus glucose importer or SGLT allow Na plus ions to move into the cell down their gradient. This inward flow of Na plus provides energy to pull glucose into the cell even against its gradient. Both Na plus and glucose move in the same direction or symport. Significance: enable cells in the small intestine and kidneys to absorb glucose efficiently even when glucose levels outside the cells are low. This image explains primary active transport, where substances move against their concentration gradient, energy from ATP. They use energy from the ATP. Primary active transport mentions that this process requires active transport. An example is given: Na plus K plus ATPase or sodium potassium pump, and there is a diagram of the Na plus K plus pump embedded in the cell membrane. Ultimately, overall, this diagram shows that 3 sodium ions are pumped out of the cell. At the same time, 2 potassium ions are pumped into the cell. The pump uses ATP as the energy source, which is indicated as blue-black arrow pointing toward the pump. And overall, the image illustrates the mechanism of primary active transport through the sodium potassium pump. Transport across cell membrane. This image showing the secondary active transport. It's a process of active transport. Here one solute, usually Na plus, moves downhill, and energy of this downhill movement is used to move the other solute uphill. Co-transport or symport and counter-transport or antiport. Its examples include Na plus glucose co-transport and Na plus Ca2 plus counter-transport. The image showing the Na plus glucose co-transport and the Na plus Ca2 plus counter-transport. This is the table showing the difference between primary active transport and secondary active transport. What is the passive transport? Passive transport is basically the movement of ions or molecules across the cell membrane without using energy. Unlike active transport, it doesn't need ATP because the movement happens naturally, driven by the tendency of particles to spread out or increase in entropy. An easy way to remember: active transport is like doing exercise, it requires energy. Passive transport is effortless, it needs no energy at all. The process showing the simple transport, which is a example of passive transport. Types of passive transport: simple diffusion, when molecules move directly through the membrane from high to low concentration, is called simple diffusion. Facilitated diffusion, when molecules move through special proteins in the membrane, still from high to low concentration, no energy is needed. Filtration, movement of molecules forced by pressure, example in kidneys. Osmosis, movement of water molecules across a semi-permeable membrane from low solute concentration to high solute concentration. Simple diffusion. Simple diffusion is a natural energy-free process where substances move from a region of high concentration to low concentration until balance is reached. Example: gas exchange in lungs. Oxygen moves from the alveoli or air sacs in the lungs into the blood capillaries because the oxygen level or partial pressure (PO2) is higher in the alveoli than in blood. Being a small non-polar molecule, oxygen can easily diffuse through the thin phospholipid bilayers of the alveolar and capillary walls. Carbon dioxide. Carbon dioxide moves in the opposite direction from the blood, where its level PCO2 is higher, into the alveoli. Like oxygen, carbon dioxide is also small and non-polar, so it diffuses directly across the membrane. Key point here: the simple diffusion requires no ATP and depends only on the concentration gradient. This image showing the transport across cell membranes, that is simple diffusion, which is the example of a passive diffusion, it directly across the membrane. Example includes oxygen and carbon dioxide. Here oxygen moves in and carbon dioxide moves out in this process. This is the image showing the simple diffusion without a helping protein or helper protein, and this is facilitated diffusion using a helper protein. Facilitated diffusion, when some molecules and ions cannot cross the lipid bilayer directly, they still move from high to low concentration, but they need help from special proteins in the membrane, either carrier proteins or channel. This process is also passive, so no ATP is required in this process. Examples: glucose transport. Glucose, the main source of energy, enters cell through protein carriers such as GLUT transporter. It moves from blood higher concentration into cells or lower concentration, where it is used for energy production in glycolysis. Sodium transport. Sodium ions move from the extracellular fluid or high sodium into the cytoplasm or low sodium through sodium channels or carrier, and it is driven by the electrochemical gradient. Potassium or K plus transport. Potassium moves from the cytoplasm or high K plus to the extracellular fluid or low K plus through potassium channels, again following its gradient. This is the difference between glucose transport, sodium transport, and potassium transport. This is the image showing facilitated diffusion. This is another kind of passive transport and requires some carrier protein. This is its peculiarity, and the carrier mediated transport, example glucose transport in muscle cells. This is the table showing the differences between simple diffusion and facilitated diffusion. Ion channels. In 1955, Alan Hodgkin and Richard Kien proposed that cell membranes have ion channels, openings that let specific ions move across. These channels are selective. Only one type of ion passes and cannot be in open or closed states. Their opening and closing are controlled by different signals, making them gated. Types of gated ion channels: voltage gated channels, open or closed depending on the difference in electrical charge across the membrane. Ligand gated channels, open or closed when a molecule binds to them. Example: acetylcholine binding to cation channels. Some respond to ligands on the outer surface like neurotransmitters, and others to ligands on the inner surface like cyclic AMP. Mechanogated channels, open or closed in response to mechanical forces such as stretch or pressure. Examples include channels in the inner ear that open when tiny hair-like structures or stereocilia move in response to sound vibration or head movement. This is the image showing the table in the differences between the voltage gated channel, ligand gated channel, and mechanically gated channel. So in summary, the plasma membrane is not just a barrier, it's highly dynamic gatekeeper, whether through simple diffusion of oxygen, glucose uptake via facilitated transport, sodium potassium pumps powering nerve impulses, or LDL endocytosis regulating cholesterol transport across membrane, is at the heart of cellular life. If this video helped you understand transport across the plasma membrane, don't forget to like, share, and subscribe for more clear and engaging biology lessons. See you in the next class. Thank you, my dear students. All the best.